So yeah, so I'm Samin Ishtiaq. I'm research engineer at Microsoft Research in Cambridge. Um, I've actually got my background is in, because of this paper you refer to, my background actually is in programming languages, in verification, in logic. And how I came to biology is kind of, I guess, the subject of this talk. And what I'm hoping also is that, I mean, you're all computer scientists here, functional programmers. What I'm hoping also is that you'll get to understand some biology as well, okay? Because there's kind of a whole new thing happening in biology. I've heard some talks about it. There's very other things happening at different levels from different aspects. So my talk is kind of is a bit different from the biology talks you've listened to so far. So yeah, so proving things about biology. Proving that's my background, verification, program analysis. Biology is kind of a fun subject nowadays. So let's go. Okay, so any biologists here? Okay, so can you kind of leave the room, or because I'm gonna pretend to be a biologist. And I once gave a talk like this, and I said, any biologists here? And somebody said, well, I'm married to one. I said, oh no, you have to leave the room as well, because that's pretty bad. So very basic biology. And what we're going to do, I'm going to show you. I've got this running example of skin. So we all have skin. This is a picture of skin. Skin is, so this is kind of a 3D model. Skin is made up of, mammalian skin is made up of five layers of cells. They all come from the same stem cell, from the same kind of phenotype. The bottom one is eventually programmed to carry on dividing and pushing up. And the top one is programmed to, so this is kind of the bottom one, the dermis. The top one is programmed to carry on shedding. Okay, so this is our skin. We're continuing, it's a growing organ. It's a regenerative organ. It carries on kind of dividing at the bottom and dying at the top. So we always get fresh skin. So what's important in a regenerative organ like skin and other things like blood and things like that as well is what biologists call homeostasis. So homeostasis, complicated Greek word, essentially just means stable. Okay, so our skin is stable, right? We don't, okay, we'll see pictures here. So what does stability mean? Stability means that the skins lost at the top amount to, the rate at which they're being lost is the same at the rate at which the new skins are being developed and at the bottom. So that's good. That's a stable position. Okay, so our organs are stable. But what if they're not? So what if we lose more cells than we make? Well, that's pretty bad, because that's what happens to older people. When they injure themselves, they have sores, and these sores heal very slowly because they're not making skin the same rate at which they're losing it. What about the opposite? So I'm describing two cases where the skin is not stable. What about the opposite? When more cells are made, so the underlying dermis is splitting and dividing all the time at a higher rate than the top one, well, that's pretty bad as well, because actually that's what we call cancer. That's a tumor, right? And look away, that's kind of a picture of a tumor. So that's going to be our running example. Okay, so we're going to see from what's called a systems biology point of view. Okay, so now we're going to try and model biology as a system. Okay, it's a very high-level system. Sorry. I think some of it. So like leukemia, some certain types of leukemia can be thought like this or modeled like this. So what we're going to try and do now is to try and understand biology, this kind of artifact, these kind of cells put together in a system, as essentially as some kind of computer program, say, okay, or some kind of artifact that we, as computer scientists, are familiar with. Okay, so there's a whole kind of new area, maybe set up in the last 5-10 years, called systems biology. And what these guys do is, previously, if you go back 20 years, even now, of course, if you go back, biology and chemistry were modeled using PDEs and ODEs, the rate of change of things. It's like when we as computer scientists, we're modeling silicon as quantum mechanics, okay, at the quantum level. But actually, that's not how we program silicon. We program silicon in a programming language called Verilog, or lower than that, we program at polygon level. We take a systems view of things, a very deterministic, programmatic view of things. That, I think, in my mind, this is what these people are trying to do. They're trying to think of biology as a system, as almost like a program. And then you can say, okay, what can we prove about this program? Can we run simulations on this, over this program? Okay, so it's a very high-level view of biology, or a higher-level view than what we're used to. So in that view, now let's go back to skin. This is a model of skin that systems biologists came up with, I think, 4 or 5 years ago. And we've got an interesting story. I've got an interesting story to tell about this model. So I've just kind of turned it by 90 degrees. So this is the bottom layer of skin. This is this is just a 1D model. So this is the bottom layer, 2, 3, 4, 5. And they're pretty symmetrical because, and that's kind of evidence from the fact, not evidence, so this is kind of symmetrical because they all come from the same stem cell. So they look the same. They have the same proteins, these variables. I'm going to call them variables because to me, this is a program. Okay? I'll explain how I see it as a program in a bit. So these variables are proteins. They're communicating, so they're signaling to each other along these lines. I've drawn these lines, of course. These lines don't exist in our skin. And they signal using chemistry. Okay? But what do I see? Well, I see some kind of communicating network. Okay, so where these proteins are just variables. The arrows between them are how they update each other. And how it computes, well, that's kind of maybe something we have to make up in order to understand whether this could be a good model or not. Okay, so let's just, so I was going to say that they look the same because they come from the same stem cell. And it's what happens in these kind of systems is that they start from the same stem cells. As they're signaling to each other, as they're evolving, as they're signaling to each other, as their fate gets determined, it's their position vis-a-vis each other, and it's the concentration of the signals versus each other that determines how they end up, that determines their fate. So this is actually a biology technical term, cell fate determination. So they came from the same place, same kind of same cell, and that will behave differently from that because they have their fate is different. Okay, so that's just a kind of a side thing on this. Okay, so coming back to seeing this thing as a program, well, okay, these are variables. This variable is updated by that variable. So wint x2 is being updated by wint 1. The circles I've drawn out around them, they're cells. So what we see is these cells have proteins inside them, and they communicate within each other and over cell boundaries. And again, this is just a 1D cutoff. This skin, of course, is a 3D, very highly complex thing. So they are all the same variable. If you see, I mean, I've just indexed them only to say that that's cell 0, that's cell 4. But they have the same signature or structure, yes. So if they're variables, but what do they look like? Well, in chemistry, there's a varying level of kind of concentration of. So I'm just making this up now because I know none of you are biologists apart from those two guys there. So there's kind of a varying level of concentration, and normally this is kind of some kind of sinusoidal wave. But what do we do as computer scientists? We go, oh well, I just chop it up, discretize it. Right, there's 0, 1, 2. 0 is off, 1 is low, 2 is high, and 3 is on. Okay, so we can model this kind of thing. In this, actually, what happens with skin is that it's model each of these variables lives within a small integer interval. Okay, so again, if you are a programming languages, programming analysis person, now you should start thinking, oh, interval domain in program analysis. Okay, and this is really our motivation in coming to this thing and trying to analyze models like this, is thinking in terms of program analysis, in terms of abstract interpretation. So these things, they have their type, or there is essentially this range, over this range, small discrete integers. How do they work? You know, if it's a program, how does it compute? Well, each of these variables has what's called a target function. The target function tells the variable what it's going to be aiming towards, right? What value it's eventually going to get. But it can only do this in steps. It can step up one in one cycle, step down in the next cycle. And I'm saying cycle now, but actually I don't mean that. Okay, I'm going to say something a bit different in a bit. So let's look at delta x1. This is its target function. It says that it's written in a very small expression language. And it's saying that, oh, if my inputs, well, 2 of my inputs are greater than 2, then some value, then I'm going to update myself, increment myself by 1. Otherwise, if it's something else, then I'm going to decrement. Otherwise, I'm going to stay what I was. Okay, so all of the target functions are written in this form. They say, is some condition met, then I'm going to increment myself by 1 or by 2. I can only step towards what my target's going to be, or away from it. Okay. So we didn't come up with these. This is something, these pictures and these formulae are something that biologists have come up with. I'm just kind of, in some sense, post kind of hoc rationalization of them is the story I'm telling you. This is what they've come up with. It's amazing because what they can do in biology now, they can almost like run a debugger on it and snapshot every state as an experiment is being run. So they have whole loads of data of what happened at every tick to every, you know, the concentration of every protein. And then they have this whole data, and then they kind of, this is what the biologists do, the system biologists guys do. They scratch their heads and go, I think this guy is related to this guy, like this. So this expression language is not ours. It's their DSL almost. That's what they come up with. Talking about how these things run, well, so this formalism is called qualitative networks. And in qualitative networks, these things can take, variables can take an arbitrary kind of integer interval. There's a precursor of these things called Boolean networks, which can only be on and off. But qualitative ones give you much more expressive power. So this is kind of something we work with a lot nowadays. How do these things run? Well, you could see them, maybe there's a global clock. Nobody believes that. But that's a simplifying assumption that you can have to do some kinds of analysis or to do simulation on them. Or we can say, okay, the whole thing's asynchronous. Everyone's just firing off and seeing what's happening. And I think that's a better semantics. A lot of what I'm going to talk about assumes synchronicity, which is actually the wrong, I'll say up front, this is the wrong assumption. But the asynchronous stuff is harder, and we're still working on that. Okay, so stability, I said homeostasis. Well, how about proving stability of these things? Okay, so what does stability mean to us as computer scientists? It means that equation, right? That single unique fixed point, okay? And here it is, written in LTL. Who knows LTL? Okay, so LTL is linear temporal logic. I'll just read it out. It just says, for all variables, there exists a unique value. And eventually, always, FG is the LTL. It's a modal logic, a temporal logic. It just says, eventually, always, those variables will have those values. Essentially, we're saying, if you consider this system, and I set it running at any arbitrary state, so this thing has no start state, no initial state. All states can be considered to be initial states. I set it running. Does it have this property of having a unique fixed point? Okay. And how can we characterize those, this kind of, the ultimate kind of, what it becomes, the ultimate states of it. So if you think of it, it's a finite state system. Everyone has a small discrete range, but there are loops in here. So it's infinitely recursing. Okay, so this really, if you think about it, for like, scratch your head for 2 minutes, you'll find there are really only 3 final states we can have. There's this one. So this is some stuff happening, some state transitions happening. I just drew a cloud there. But what happens is that eventually you come out, and there's a single state, and you revolve around that. So your next state is always that state. This is the single unique fixed point. And for skin, that's kind of what we're aiming for. But the other ones are interesting as well, and we just need to kind of say, these are different kind of characterizations. These are different kind of final outcomes of a system. So here's one. So this is not a single. Sorry, this is not a cycle of of length 1. This is a cycle of more than 1. So this is actually what we call a cycle. So we come to some state, and then it takes at least 1. So I've just drawn 2 here, 3 here, but at least 1, and so we cycle back. So that's another state we could have. Bifurcations. So we're doing some stuff, and then there are actually 2 unique fixed points, or 2 cycles. But the state bifurcates, and it stays like that. OK, so let's go back to it. OK, so how are we going to find stability? I think the way we've come from it is thinking of that system as a graph, thinking of how can we do abstract interpretation on it in this kind of interval domain. And there's tons of work on this. And what's really been interesting is that all those algorithms and all those procedures of program analysis that have been attempted to be applied to real programming languages. So one of the projects I work on is trying to do abstract interpretation on C for Windows device drivers. That's really hard. So a language like C is so difficult to analyze. It's really hard. But here, we've taken really something quite simple, a really simple program analysis, and applied it to these programs, which are really simple and kind of almost stunted programs. And they're almost low-hanging fruit for program analysis. We've just taken them and found things out. So this is a very simplified version of it. What do we do? And I'll show you a picture in a bit, which will explain what's happening there. So we take the network. We generate lemmas in LTL. We generate lemmas to tell us what's really happening. And what we're hoping is that we can put all these lemmas together and eventually find that final formula that we wanted. Okay. So what are we going to do? We're going to generate some lemmas off the network. We call that bounds, those lemmata. And if somehow from those lemmas we can say we're stable, then we return stable. Otherwise, there must be a counterexample somewhere. It's not stable. So then we hunt for those other states, the cycles. Yep. Sorry, this is. I don't know. It's just something to talk about afterwards, I think. Okay, so and the kind of the crux here is how do we do this? Okay, that's just okay. How do we do this? So this is kind of where I struggled a bit because I really wanted to take my implementation, which is in Fsharp, and show you that. And it's purely functional. It is a recursive function which goes through until it finds a fixed point. But the more I tried to write it down on one slide, the more difficult it became. And in the end, I thought, OK, I'm just going to talk to a functional programming audience using this high-level imperative algorithm instead. It's actually easier to look at. I don't know why. The functional one, which is recursing over itself, taking some state around, trying to find the fixed point or something is really difficult to patch down into one slide. So what's going to happen? So like all kind of program analyses, we're going to have, what we're doing is we're going over the state space of this network. And we're starting somewhere, we call it the frontier, F for frontier. And we're going to start with all the, essentially the non-constant variables. And we're going to carry on analyzing them, analyzing their outputs, until we've reached some kind of quiescence. Okay, and that's what this kind of, this procedure does. It picks a new variable from the frontier, generates some lemmas for them. And I'm making up notation here. I'm adding lemmas to these predicates that we already have. I'm just making up notation just to kind of get the idea of what's happening. So we're just generating new lemmas for that guy and saying, if I've got more information, then carry on working. Add it outputs to it and carry on working. Let me show you a picture of this. So here's our skin model. We're starting, sorry, you can't see, and I'm standing in the middle as well. So we're starting, say we start here arbitrarily. We're actually starting in this source node, which happens to be constant. So this is kind of a good place to start. But I could have arbitrarily picked somewhere else. We start here, and we can prove that this guy is always positive. And using that, we can go there, because these are its outputs. We can prove something else, we can prove something else, we can prove something else. What we're doing here is we're kind of doing a lot of local searches, and we see the proof kind of rippling out. And we're finding kind of, starting from islands of stability, we're spreading the knowledge we have until we know all of the thing about the network. Okay, and that's all very well, and we had an implementation of this using, a bunch of theorem provers, like automatic provers like SMV and things like that, but it was quite slow. And actually we realized that we don't have to be so general. Why are we looking for lemmas of a particular shape? Why don't we look at kind of the interval domain that we have and try and work on that? So what does stability mean in terms of intervals? Well, all these guys begin with interval 0 to 3 or 0 to 4 or whatever. Well, what if we could do some kind of analysis to say, to narrow these intervals down until they're singletons? And that's kind of the same as saying you're stable now, because we're saying that you're now just this one value. You can't be between 0 and 1 and 2 because you're only 2. That means the whole network. If we can do that for all of the network, narrow the range down to singletons, you're stable. The network is stable. So it's not the same as finding termination for this kind of thing, which is, like if you go several slides back, I was talking about, is there a unique fixed point, which kind of amounts to termination a bit. But now we're saying, can I just narrow your ranges down? And there's a very fast way of doing that. And we have an implementation of it in, 40 lines of Fsharp, which does that. What does that do? Well, let's go back to gen lemmas. I've called this faster this time. And all this does is calculates new bounds for W. Okay? And it does this by saying, okay, W, who are your inputs? What are their current bounds? Let me just symbolically calculate your new bound. Is it less than what it was? Then yes, I'm progressing. So I'm going to add W's outputs back to the frontier set and carry on. Okay, so this algorithm runs really fast. So we have QNs with thousands, tens of thousands of variables. And this is kind of an online, I'll show you a screenshot afterwards. This is a tool running on a website. People just click the button and it says, you're stable, you're not stable. Well, sorry, it says you're stable. Instability is a different question. Okay, so let's go back to the original outer algorithm. Right now, it's kind of essentially a one-shot thing. So it says, here's the network, run the algorithm, here's the result, I'm done. What's actually nicer is that what's actually nicer from a debugging point of view, and also from the point of view of people who are using this tool to understand how the proof is spreading around, is to say, can I actually have the values at every tick of what's happening? Okay, can you give me essentially the bounds or some kind of, the thing that's leading up to the bounds? Okay, so like a history of what's really happening. I mean, if I kind of use the analogy of debugger several times, what's really happening as the proof is going out? I want kind of a snapshot at every time. Okay, and there's an easy way to, well, not an easy way, but I can take this code, take this this algorithm, and get it to do sequences, return sequences instead, sequences of results. Okay, and so right now I go from QN to result. This one, it kind of takes, you have to just sit and stare at it for a while and see what's happening. This is the outer one there. The inner one, Gen lemmas, its type is different now as well. It returns a sequence of results. And this guy is constantly outputting the current state until it's done, and then it outputs the final state. Okay, so this is really useful because essentially the user can now go tick, 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 fast forward until 20 steps, stop now, go back. Okay, you kind of have some kind of idea. The user could be in charge of seeing how the proof is flowing out. So this is a kind of really useful thing to say, take my one-shot algorithm and turn it into kind of some sequence thing instead. Okay, what about instability? Well, in cases where it doesn't work, what do we do then? Well, we try and, where it doesn't work, what do I mean by that? I mean, we haven't found this single unique fixed point using this kind of idea of narrowing down the intervals. If we haven't done that, then, well, we hunt for bifurcations first. If those aren't found, we hunt for cycles. If those aren't found, we hunt for fixed points. And the algorithm is sound, apart from the fact that we might be losing some information because of the abstraction we're doing. Okay, so this thing in particular, so these are all done. So the algorithm I talked about previously is a kind of a custom algorithm implemented on these QNs. These procedures take the problem of finding bifurcations and cycles and encode them into SMT problems. Okay, so who knows about SMT? I guess some, okay. Okay, so those who don't, this is an ideal time for me to say, I'm giving a tutorial tomorrow on SMTs. So come to that. Okay, so SMT stands for, actually somebody referred to it this morning, I think, stands for satisfiability modular theory, thank you. So it's a kind of essentially a black box solver. You give it a problem in propositional logic plus a bunch of theories. You could have arithmetic, linear arithmetic, you could have quantifiers. So I started with propositional. You could have quantifiers, you could have arrays and things like that. But it's a black box. You give it the problem and it'll say whether it can find a satisfactory model for it, or it can't find a solution for it. Okay, so these procedures are all encoded as SMT problems and given to this black box. Bifurcation is is an ideal problem for SMT encoding, because you say find me a fixed point, and the SMT black box, in this case Z3, comes back and says, here you are, and then you say to Z3, okay, don't find me that one, find me another one. So you negate it. You say, I don't want this one, give me another one. Can you find me another one? And it says, yes, here's another one. And then you know, then you bifurcate. So that's a really nice problem to code up in SMT. Cycles aren't. And again, over the last year, we've been thinking of how to solve that kind of intractability there. And we've come up with, again, with some custom algorithms to do that. I won't talk about that too much here. Okay, so that kind of deals with stability, instability, okay? And so essentially it's saying, can I find this unique fixed point can I find these other kinds of stable states? What about general properties? Okay, what if I wanted to say, what initial conditions, does this initial condition lead me to a fixed point? Does this, if I have a fixed point, what could my initial conditions be? Is it true that for this qualitative network, after this goes high for 2 cycles, this goes low eventually, always goes low afterwards? What about those kind of properties? So those are interesting as well. And we have a kind of an implementation of an LTL checker. So you can say these kind of properties, as long as you can say them in LTL. We have a kind of an implementation of an LTL model checker for these qualitative networks. So LTL is kind of essentially a very, the smallest core of LTL takes propositional logic and extends it with these 2 operators. X means next. So you're saying XP, P will be true in the next time tick. Until is an operator which says that something is true until something else is true. Okay, F until G. And using these 2 operators, you can code up a bunch of other things like F and G and things like that. So these are kind of the most primitive ones of all. So how do we code this up? Well, actually, we go back to an idea from a long time ago. Well, actually, I think it was published first in 2006, but I think Amir Noeli's idea of temporal testers, which goes back. I think his idea goes back before 2006, and the idea is to take LTL formulae and to have Boolean variables which track each subformula. And then you can code the whole LTL problem up as a SMT one. Okay, so again, we're kind of saying Z3 to the rescue again. We're taking LTL formulae, coding them up for Z3 to solve, and kind of putting some model checking in there as well. And Z3 will give us the answer there. So there's some other things we have to code up. So Noeli's temporal testers idea, this idea that we have Boolean variables to track the truth of temporal formulae, is not enough because we need some idea of fairness as well. And that we just encode on top using Z3, using SMT formulae as well. So I just want to give you, how are we doing for time? Just want to give you an idea of some of LTL. So here's an LTL formula. Next F, so it says that, I should have lent a bit more space here actually. So it says XF is true in some state S and at time t if at the next time period F holds. So I've dropped the X because I'm in the next time. Okay, so how do we encode this up in using the idea from Noeli of temporal testers, where we have essentially we have variables tracking this guy and this guy. And the variables are kind of indexed by time, and we have to encode a lot of bureaucracy into that we have to give to Z3. We have to tell it, this is time, this is the next time, this is true then, that now this is going to be true next. So I have to put in a lot of bureaucracy into Z3, a lot of kind of bookkeeping, and that on top. And that's tracking the idea of next F. And F until G, this is what I'm showing here previously as well. This is just the normal semantics of LTL formulae. So it says that, so F until G is true if there's some time. So time here, you see time as a linear thing. There's another logic where time branches. That's even more fun. So there's some time later than t, and at that later time, G is true, and all the time before that, F was true. Okay, this is just the normal LTL semantics. I'm not making this up. So this is just linear time. Yeah, but so LTL is linear time. CTL is that branching one. In CTL, branches don't merge back. It's kind of just a tree. But this is not CTL. And this is encoded like this. And if you think about it, actually, again, if you squint your eye a bit and look at it, this makes a lot of sense, actually. So it says that the Boolean variable tracking this guy, if that's true, then either G holds this guy, or F until G, F until G. So F is true until G is true. F is true until, this is kind of the recursive part of it. So F is true until G is true. So I have to squint at myself, I think, on a piece of paper sometimes. Okay, I can skip over this actually, I think, but we'll see how we go. So this is kind of a function programming conference, so I have to have some kind of theorem or some kind of logic thing going on to scare everybody else away. So these systems, but don't worry, this is the only page of a theorem. The next thing is pictures about this theorem. So what's true in these kind of systems is that if I can't reach S prime, if I can't reach it after I steps, right, then I can never reach it after more than that many steps. Okay, and that's kind of, if you think about it, that's kind of a obvious thing. In these kind of systems, you're kind of, you're kind of chuntering along, right? And if I could never get there, then I can never get beyond there either. Okay, because I'm still here. Okay? If I can never get there, I can never get beyond it. Okay, so it's kind of an obvious thing, but actually it's not that obvious as well. But what it lets us do is cut down the search space a lot. Okay, so this is an idea that came to us much later than the original stability algorithm. The original stability algorithm doesn't use this. The LTL checker does use this, and actually it's a kind of a work of mine to go back and say, I really need to put this back into the stability checker as well to optimize it. But what this does, what does this let us do? It says, so here's like some 2D shape I just made up. Say this is the search space of our QN. That lemma there, what does it let us do? It allows us to chop this down, okay? Chop it down, chop it down, chop it down, until we're left with much smaller space. This is what the LTL checker does before it runs the LTL check, before it does the LTL thing itself, check itself. It cuts down the space. And again, this is kind of something that we came up with over the last year. Okay, so enough about theorems and stuff. How are we building this? So we've got biology, we've got real biologists saying, oh, can we have something like this? So we're going to build a tool on it. And there was an interesting story. I should have told you this right at the beginning, actually. There was an interesting story about the skin model. When we were building this tool, we said, oh, what should we do as an example? And somebody said, oh, let's do skin. It's kind of, there's a known model for skin in the literature. So we coded it up. And we ran our stability checker on it. And it said, unstable. And I went, oh, no. Who wrote the code? Oh, sorry, it's me. So looked at the code. Actually, there was nothing wrong with the code, it's turned out. And, well, no, rather, actually, at that point, I thought, no, there must be something wrong with the code. So I was looking at the code. And Jasmine, the biologist in our team, said, actually, no, there's something else going on here. And she was kind of scratching her head and looking at the model and said, the model is wrong, actually. And it turns out that the model that the biologists had published in the literature, and everyone thought, oh, that is the model, that is how skin is, was wrong. There was some bits missing of it. And our tool had actually found a bug in that model. Unluckily for us, literally in that week, some biologists had done some experiments and also found out. And so they were kind of first to publish and first to say, oh, that thing is wrong. But it's very interesting that we were working on this tool and we actually found a bug in biology because of that. And the bug was actually really simple. There was literally one influence arrow missing in each cell. There are two variables. If I needed to draw one more arrow between them, then the whole thing would stabilize. Without those two arrows, and I haven't shown in that the ones I've shown you is a stable version. Without those arrows, 5 arrows, it doesn't stabilize. So it's kind of you always need to write the code to prove some idea, right? And in doing that, you'll find something out. Okay, so what have we built? That's kind of all the implementation of these algorithms in Fsharp. At the top, the whole thing lives on a website as a web app. The whole thing on the front is the UI written in TypeScript. So I noticed at the talk after me, maybe it'll inspire me to say, oh no, ditch TypeScript and do it in something else. So that's all written in TypeScript. It just exposes, that talks to a REST interface in .NET. The Fsharp code sits behind that and makes calls to Z3. So again, what I've told you about is that there's some custom algorithms. So stable is a custom one. And these guys are not that special because they mainly call Z3 to do their work. And again, if you want to find out about Z3, come to my tutorial in the morning. Okay, so I'm by background, so I'm just going to talk about some general stuff to do with functional programming. My background is actually here, from university and things like that. I'm used to using Emacs, and I'm used to using Camel. Actually, from one of my other big projects, I use Camel a lot. And this has been a really interesting experience to use Visual Studio and Fsharp. And I think they're actually though they are functional programming languages both, and they both share a very common core, maybe because of the editor, maybe because of how it does its type checking, Fsharp does its type checking, they're actually, the way I think when I'm programming is very different. So I don't know people who do functional programming in different programming languages, how they feel about picking one versus another one. For me, this is a very interesting contrast. And I find I have to think more before coding in Camel, whereas in Fsharp I can have a bit more fun because I can just explore as I go along. Okay, so this is the tool that we built. Just a screenshot of it. If you search for Biomodel Analyzer, you'll hit an old version of it. This is kind of a testing one that has the TypeScript front end. I always say, whenever I show this, whenever I show this, I say, I'm the computer scientist. I'm the guy behind this button. Okay, so there was so much work done into this tool, and it looks so beautiful. And it is what, when I show this demo this to biologists, they go, oh great, I can draw a cell, I can draw these different variables. One lives, this one always lives on the boundary. This one always lives outside, so it kind of models the environment in which your model lives. A biologist knows exactly what to do. When I show it to computer scientists, they go, well, first of all, they draw some kind of 2-variable oscillating network, and then they go, what next? And I go, well, it's not meant for you. You draw your 2-variable oscillating network, and that's the end for you, right? The biologists, they immediately understand. This is what they normally draw. They publish papers having drawn these things in PowerPoint. But these things are then never usable from PowerPoint. They're like static, drawings on a page. Whereas this, you can give this to somebody. It's a model. You can save it, give it to somebody, run analyses on it. So it's really interesting to think in terms of users all the time, users first. And I don't normally do this, but some of my co-authors do this. Jasmine, in particular, who's the biologist on our team, she always does this. She shows the team. And I think it's really interesting because a project like this one, if it was just a bunch of computer scientists, and actually just the nerdy-looking people on it, if it was just a bunch of computer scientists, we wouldn't have been able to do this. We'd have some command line thing that only ran on Linux terminals. Whereas if you've got biologists and Ben's a biophysicist, and Alex is an anthropologist, you get these people involved, and the whole project could become something different. OK, so that's all I want to say. Thank you very much.